So we're very pleased today to have with us Paul Thomas, PhD from the St. Jude Children's Research Hospital. His talk today is experimental and computational approaches for analyzing T cell receptors to identify condition-associated TCRs and determine their specificities via reverse epitope discovery. A little bit about his laboratory. The lab is interested in deciphering human susceptibility to disease and how the immune response contributes to productive or pathological outcomes. They conduct multiple types of human cohort studies with a particular focus on pediatric studies of influenza and SARS-CoV-2 to learn about the correlates of protective or pathological immunity. They then translate these insights into mechanistic studies to define the immune response and identify steps we can encourage or inhibit to impact therapeutic intervention against infectious diseases and tumors. Research interests are innate and adaptive immunity to influenza T cell receptor repertoires in infection and cancer, influenza-associated immune-induced healing responses, and pathology, and CD8 T memory cell and memory and recall potential. Just a little bit about him. Following his postdoctoral training with Nobel laureate Dr. Peter Doherty, Dr. Thomas joined the faculty at the Department of Immunology at St. Jude in 2009. His work has been instrumental in generating new understanding of T cell receptor specificity and defining key elements of innate and adaptive immune responses that determine clinical outcomes to viral infections. He leads several complex institutional programs, including human influenza surveillance efforts and virtual repository development, and currently serves as PI for the 7-year 12-institution Da Vinci Consortium. In addition to his scientific achievements, he's widely regarded for his mentorship and empowerment of junior scientists. So thank you very much for joining us today. We're really excited to hear more about your work in this realm.
Thanks so much for that kind introduction, and I'm excited to present my work to this group today. So what we're going to be talking about today is the molecular basis of T cell receptor recognition. And so this is the structural schematic of the T cell receptor here in purple, and its target antigen, the peptide MHC, in blue and yellow with the peptide. And you can see these loops hanging down from the TCR that make contact with the peptide and with the MHC. And these are the hypervariable loops, the CDR3 loops of both the alpha and beta chain of the TCR. For a lot of my work, I'm focused on anti-pathogen immunity or anti-tumor immunity. But I think the principles that we'll talk about today apply very well in autoimmunity as well. And so the question I'm going to be addressing today is how do we measure T cells in humans? How do we identify correlates of protection or severity? And in part, this comes down to trying to ask what antigens we see from a pathogen. So what peptides sit in this peptide MHC groove? This is, the basis of the activation of T cell responses, going from the naive T cell to the effector T cell. The difficulty of this in humans is, of course, the vast diversity mediated by host MHC molecules. And so when we're talking about CD8 or class 1 molecules or CD4 and class 2 molecules, in humans, we've got 6 class 1 molecules, A, B, and C, with alleles from your mother and your father. And for CD4 class 2 molecules, we have DR, DP, and DQ. They can actually share chains, and there can be additional alleles of DR. And so it's at least 6, but potentially 12 molecules of class 2. And so this creates a vast potential diversity because this is the most polymorphic region of the human genome. And so we all have slightly different class 1 and class 2 molecules, though there are some that are more shared, and there are certainly some that are more associated with particular protective or pathological responses, including in diabetes, as this group is more familiar with than I am. But those HLA associations actually point to the importance of specificity, because it means that very specific peptide targets are critical for mediating the disease. And so if we come back to this, this creates a huge problem for us, even in studying a common pathogen like influenza or SARS-CoV-2, because none of us see the pathogen in quite the same way. And so if we move to the other side of this equation, we can say, well, if we look within relatively dominant HLAs, do we at least use the same T cell receptors when we see the same antigens? So HLA-A201 is very common in Caucasian populations. DP 0401 is actually common across the world in about 50 to 60% of individuals for class 2. And so can we just rely on the TCR directly as a hook into the response? And the problem there is that there's an even greater diversity on the T cell receptor side of things than on the antigenic side of things. And this is mediated by the VDJ recombination process that generates the T cell receptor, which I'm not going to dwell on too much today. But the important thing to know is that for both the alpha and the beta chain, there's this semi-stochastic process of recombination at the DNA level that creates these hypervariable junctions that are purposely error prone to create a potential diversity of, on the very low end estimates, 10 to the 15th, this is sort of a functional estimate, up to a theoretical estimate of 10 to the 61 potential receptors. And so if recombination were fully random and there were 10 to the 61 potential receptors, then we would actually never see the same receptor probably twice in all of human history. We do see some sharing of receptors, and I'll come to that later in the context of diabetes. And that reflects the fact that recombination is not fully random and there are biases in what mutations are made at the hypervariable junctions and what gene segments are used. And so what we're left with is this great diversity on both ends of the spectrum. And that's forced sort of two modalities of measuring T cell responses to kind of take over the field, I think, in terms of how work is published in the literature. In pathogen-specific immunity, and to some extent in autoimmunity, the vast majority of papers rely on what I call bulk methods, functional-based assays. And so these include things like intracellular cytokine staining, the activation-induced marker assay, the AIM assay, or ELISpots. And the advantage here is, and so in all of these assays, you take T cells out of a person and you stimulate them with your target antigen in some form. So it can be a whole virus, it can be a pool of peptides that you associate with a particular antigen, it could be insulin protein or insulin peptides. And the advantage here is that it's comprehensive, so you can cover a whole antigen in theory. You don't have to know anything about HLA to do this, and it's relatively cheap. And so you set up this co-culture and you look for activation of these T cells by cytokine secretion or surface marker upregulation. The downside is that it's very hard to get a sense of sensitivity in this assay. We know it's not fully sensitive. We know the more complex the antigen, the sensitivity declines. And there's no real gold standard to measure it against. And so it becomes somewhat self-reinforcing that if lots of people do the assay is seen as the gold standard. And specificity is potentially even worse. There's quite a lot of background. And in the context of pathogen-specific responses or autoimmune responses where somebody is baseline inflamed, you have the problem that you're looking for a delta from that baseline to measure your activation. And if they're already activated, you're not going to measure that cell as a specific cell, and you could be undercounting. On the other end of the spectrum, we have highly sensitive and specific methods using multimer technologies. So these are peptide MHC multimers, the most common form being tetramers, but there are dextramers and pentamers and various commercial applications of this, where you have a fluorescent tag on a complex of peptide MHC molecules. Again, these are very specific, but they are HLA restricted. So you need to know the HLA of the person that you're looking at. And you have to have mapped an epitope, and you're only looking for the things you're looking for. And so that if there's an important response in that antigen that you didn't know was an epitope, then you don't scan for it, and that information is not acquired. They're also pretty expensive. So doing, anywhere from maybe 400 on the low end to, several $1,000s on the high end for even one of these reagents. And so if you imagine there's 50 to 100 epitopes per pathogen or per complex autoimmune disease for a single person, to get comprehensive coverage across the population is currently functionally impossible. So what my lab is focused on is to try to fall back on our understanding of T cell receptor biology and their target antigens to try to find a way through this paradox. And so is there a third way where we can do these assays that will get us the sensitivity and specificity of tetramer or multimer reactions with the low cost and high throughput of a bulk method? And our approach arose out of observations we started making years ago when we were looking at T cell receptor sequences coming out of mouse influenza infections. And we were not the only people making these observations. which was that while you rarely saw what we would call truly public receptors, meaning that the exact same sequence on the alpha and beta chain are found in multiple animals, you would see highly similar sequences. And you could actually train yourself to get quite good at recognizing these similarities. And so these are 3 sequences that target the same antigen from an influenza-specific response. And so this is the alpha chain CDR3 hypervariable loop and the beta chain CDR3 hypervariable loop. And you can see there's very few differences here. They use the same J beta. And even this third chain that is actually quite different. It uses a totally different alpha. It uses a different V beta, but it does use the same J beta again. So this SAETL is conserved across almost all of these responses. And again, we could, because this was years and years ago and we were sequencing at a relatively low throughput, we would sort of hand curate these sequences and you would learn associations with specific epitopes. And so we decided to take it out of our intuition and turn it into an actual metric where we weighted the similarities and differences among these different hypervariable regions and accounted for the fact that the CDR3s could be different lengths and have variable substitutions across the full length of the CDR3 loop. And that metric that we developed is called TCR dist and was published several years ago now in collaboration with Phil Bradley. And it's really a side-by-side comparison of any 2 T cell receptors. And so this is just a T cell receptor depicted structurally again here with the surface that makes the interactions with the peptide MHC highlighted in color. And we just have a weighted score of the similarities and differences based on a BLOSUM 62 matrix across that entire sequence. And this is actually a really simple metric compared to many iterations of similar metrics that have come out over the years. But it still ends up competing quite well in head-to-head comparisons with even kind of complex machine learning based approaches. It usually finishes in the top 3 and it is far less computationally intensive. And so if we look at what we can use that score for, if we take those same TCR sequences I was just showing you on the previous slide and now take 800 sequences that target that same epitope, which is an epitope derived from an influenza protein, we can order them now in a quantitative manner on a hierarchical clustering tree where we can derive motifs from clusters of closely related TCRs. And so in this bottom cluster here, there's 254 TCRs that have this motif. This is the V alpha region that's used, the J alpha, the V beta, and the J beta. You can see the V beta is quite flexible. That J beta 2,3 is conserved all the way up the tree. There are heavy conservations on the alpha chain as well. And you can see the regions that are allowed to be variable. And so we've now used this core insight that T cell receptors that see the same antigen look very similar in a way that can be quantified and put and statistically given some confidence in terms of how similar TCR has to be to likely see the same antigen. And we've used this to develop this pipeline that we call reverse epitope discovery. I'm going to show you a published example of this from SARS-CoV-2 where it worked particularly well, where we were in the situation where the field was moving very rapidly. Very few epitopes had been mapped. And in fact, when we started the project, I'm going to show you no class 2 epitopes had actually been mapped. And we wanted to generate a multimer tool to track vaccine-induced responses in human draining lymph nodes for which we had very, very small amounts of material. So that meant that we were unable to run any of the classic bulk type assays, these functional ICS-AIM type assays. And so we needed an actual antigen-specific target. And so what we started with was the TCR sequencing, and we were going to work from that TCR sequencing to infer an epitope and an HLA allele, and then validate it by cloning and expressing a TCR, an HLA allele, and an antigen to show that we had identified the right interaction. And this was done in collaboration with Ali Eliabetti at WashU, who had really developed and mastered this technique of doing fine needle aspirates on humans receiving either the flu vaccine or the SARS-CoV-2 vaccine. And we're sampling the axillary lymph nodes that are inflamed after the vaccine here. And you can see the needle going into the swollen lymph node. And so he had published a lot of work on the B cell response to this vaccine. This was the BioNTech mRNA vaccine. And what he sent us was RNA sequences from 4 individuals that had received the vaccine where he had done bulk TCR sequencing. And so bulk meaning it was unpaired, and we had just a list of beta chains and a list of alpha chains. And what we did is we plotted the beta chains from these 4 individuals in a TCR space defined by that TCR dist metric that I showed you a few slides ago, where TCRs that were within a radius that we thought were likely to target the same antigen are connected by an edge in this map. And they're colored by the person that they came from. And so what we were looking for were clusters of TCRs. Each circle here is a unique TCR, so we removed clonal expansions. And we were looking for clusters of TCRs from multiple individuals that were connected by multiple edges. And so this cluster right here is a good one. It's in 2 people. There's multiple TCRs that are converged on likely the same antigen. And so that would be a good candidate. And I always show it this way because a lot of folks do just TCR beta sequencing because it's commercially available. But if you also ran the TCR alpha sequencing on the same sample, there was a single cluster that was in all 4 individuals. The size of the circle here represents the number of edges that is connected to that particular clonotype. And so you can see that in that large middle cluster, every clonotype is connected to every other clonotype, and it has this highly characteristic CDR3 motif defined by TRAV35 and TRAV42 with basically 1 residue that could be variable. And so this is clearly a condition-associated response with no partner on the beta side that's obvious. So there was no equally large cluster. And so we inferred that this was SARS-CoV-2 specific, but we didn't know, and we certainly didn't know what the target epitope was. And so this is where we sort of turned the crank for the first time on this reverse epitope discovery approach. So we have an alpha chain motif, and we dug into public single cell data from individuals that have been infected with SARS-CoV-2. And remember, our folks have been vaccinated. But we found our alpha motif in people that had been infected, and that gave us beta chains that could pair with that alpha chain motif. So we had a list of beta chain sequences that were paired with our alpha chain motif. And again, we're not necessarily looking for exact matches because exact matches are relatively rare in the world of TCR space. So of course, with this conservation of the motif, we do find some exact matches. And the reason we really wanted beta chains was really twofold. One, if we want to reconstruct a receptor, improve specificity, we needed an alpha beta pair. And 2, if we had access to a public database from an assay called the MIRA assay, which is a functional based assay where instead of just counting the number of cells that get activated when you stimulate them with a pool of peptides, you actually sequence the beta chains from those activated cells. And so this had been published by Adaptive Biotechnologies, and they had used different pools of peptides covering SARS-CoV-2 antigens in individuals that have been infected with SARS-CoV-2. And so what the MIRA database was a list of the peptides in each pool and the beta chains that were associated with activated cells stimulated with those peptides. And multiple peptides each peptide was in multiple pools, with each peptide having a unique address in that it was in a different set of pools from any other peptide. And so what that let us do was look for the beta chains that were associated with our alpha chain. And it was notable that the beta chains we picked didn't really have an obvious motif and actually hadn't been curated by Adaptive, which had already heavily analyzed this data set, to find an associated response. And so it again gets into the fact that a lot of the specificity of this response was being driven by that alpha chain. The process here, it can sound a little convoluted, but it's a very simple combinatorial deconvolution problem, which is similar exactly to the same kind of calculations that you do when you're doing these little puzzles when you were little about kids sitting next to each other in class and reading different books and wearing different colored hats and trying to figure out who's sitting where reading what book. And so when we applied, when we looked in the different pools of stimulated cells for our beta chains associated with our alpha chain, we found one set of peptides that was associated with those beta chains. And they were from the spike protein, which was great because our individuals had only been vaccinated, so they should have only seen spike. This was really our first confirmation that this was a SARS-CoV-2 specific response. But it was a relatively, the way they set up the pooling was a little bit inefficient. And so it was a very large region of the proteome, 58 amino acids, which is much larger than an individual epitope. So we had a 58 amino acid region of spike to work off of. By this point, though, we had figured out the MHC alleles of our 4 individuals, and we knew all 4 individuals had had this response, and they only shared one class 2 in common. And the class 2 that they shared in common was DPB0401. And that allowed us to then predict a single peptide from within that 58 amino acid region that we could that could bind to DPB0401. And that allowed us to make a tetramer, which we can now go back to the samples and show specific cell binding across the time course of post-vaccination lymph node sampling. And so that's just depicted here, where you have this nice lymph node response in blue, the blood response in red. This is a log scale, so you can see the lymph node response is relatively flat in most individuals, whereas the blood response peaks very quickly and then declines down to very low levels, really at the limit of detection for flow cytometry, which is about 0.01% reliably. We could also just track this response now by sequencing, because we had a very strong motif in the alpha chain. And so what I'm plotting here are 2 different time points from each individual. So day 28 here versus day 60. Each circle is a unique TCR again, and their relative frequencies are plotted. And so you see almost everything falls on this X equals Y line, meaning that the clonotypic structure of the repertoire is quite stable over this time course. And all of the clonotypes with our alpha chain motif are highlighted in red here. And so you can see it's hugely dominant in these individuals. It's stable over this time course out to actually 110 days in this initial analysis that we did. And we can plot our different clusters of TCRs in these sort of box plots here. And in 3 of our 4 individuals, the DPB0401 S167 response was the largest response, and the 4th individual was the second largest response. But we can also infer that these other clusters are likely specific responses because they're stable in this lymph node over time, particularly these ones that are clonally expanded. We applied this method now to the next largest clusters, including that beta chain cluster I showed you. And we found that 4 out of the 5, we found 5 new epitopes, 5 epitopes that we described as the next 5 clusters. And when we published this paper, 4 out of the 5 epitopes had not been defined, despite a lot of effort across the field to define SARS-CoV-2 specific responses. And so another advantage of this approach is that it gives you the immunodominant responses directly without sort of these functional assays intervening. Okay, so what I showed you in this first part wasn't so much a mRNA vaccine story, but mRNA vaccines induce this robust and stable germinal center TFH response. But the main point that I wanted to show this was the immunodominant responses are often missed by conventional mapping and can be identified by reverse epitope discovery. And these broad pathogen-specific response dynamics can be determined by sequencing alone once you know what sequences you're looking for. And so in the second part of the talk, I want to talk about this as a more broad approach where what you're really looking for in a T cell response is this delta between a baseline response and the effector response and the convalescent response. And so you just need something to change over the course of time to capture these condition-associated TCRs. So in a classic infection or vaccination model, we have a true baseline. We have the peak of inflammation where these cells have expanded, and then we have the contraction. And if we just sample across this line, we can assume that those TCRs are condition associated. So often we don't have a true baseline sample because we're not enrolling large prospective cohorts. Sometimes we are. But so we could take a peak sample at the peak of the effector response and then a sample at resolution. Or conversely, if we do have a true baseline, we can take that sample and a sample at the peak. And this is what those data look like. This is how that works. And this is a case of sampling where we didn't have a true baseline in the graph I'm about to show you. And so when we were doing these assays initially, and I'll show you, we've updated this protocol quite recently. We would always run these bulk TCR assays in duplicate, and the reason for running them in duplicate is we would plot the two duplicates against each other. So we would take each sample and split it into two independent runs. And so you would have, in this case, an acute sample from someone infected with SARS-CoV-2 against their own acute sample, or the convalescent sample against the convalescent sample. And you'd plot all of those T cells against each other. So in these two graphs here, all their clonotypes against each other. And this allows you to create a noise model because one T cell can only be in one sample. And so at the very high end of the distribution, and you can see this is a multi-log scale distribution, and we'll come back to that. There's great agreement between the two samples, X equals Y. But as you start getting lower and lower, if you have one T cell, it goes in one tube and not the other. And if you have 2 or 3 T cells, maybe all 2 or 3 go in one tube and not the other. And so you get this curve that you can make a noise model and say, this is the natural curve of natural variation across tubes. Anything that differs from this curve is biological variation that's probably meaningful. And so if we plot acute versus convalescent, now we have all of these receptors that are much larger at the acute time point than they were at the convalescent time point. And so these are the SARS-CoV-2 associated responses. There's also cells that actually are continuing to expand into the convalescent time point. Those end up being mostly CD4s, and we think they're mostly Tregs. I'm skipping quite a lot of data for the sake of time, and since this is more focused on autoimmunity, and I want to get to some methods at the end, but we can take all of those condition-associated TCRs and now infer the overall magnitude of the SARS-CoV-2 specific response. And in this study, we actually did have a true baseline, from that baseline, we could look at T cell responses that expanded into the acute and show that some of them were relatively high at the baseline time point. And these were cross-reactive responses to common cold coronaviruses. And we went on to show that those specific TCRs reacted to specific epitopes from common cold coronaviruses. So you had this about 3 or 4 log range of memory that people had to epitopes that would be presented by SARS-CoV-2. And we and others have made arguments that people that were on the high end of this curve were actually protected from severe outcomes from SARS-CoV-2 versus if you were on the low end of this curve. At the acute time point, people sort of condensed around about 10% of their repertoire being focused on SARS-CoV-2 in the blood. And then you had a spread again on the convalescent time point. And part of this is probably just variation in our time point sampling. Over here, you can see that we're looking at just what's happening to everything that's not SARS-CoV-2 specific, inferred SARS-CoV-2 specific, and it's remaining flat. And we can also look at what's happening to things that we know what they are specific to. In this case, this is a flu-specific response, which also remained flat. And we only looked at people that had the right HLA for this flu-specific response in this graph here. One person had this very high response at baseline, and we think they were coming off a flu infection when that sample was taken. So we can now take all of those condition-associated TCRs and plot them in TCR space again and get this sort of epitope level resolution map that's similar to the maps I showed you before, but now made for many, many more people where we can work backwards and deconvolute these specific responses. And so what I've shown you in this part is that we can just do longitudinal tracking by sequencing that permits this repertoire scale detection of response magnitude, specificity, if you can do the decoding, phenotype with pre-enrichment, and cross-reactivity with or without decoding based on baseline to acute expansion levels. These SARS-CoV-2 responses were a variable magnitude across individuals. They could reach up to 30% of detectable T cells in the blood. You don't deplete your baseline repertoire. So those big clones that were not SARS-CoV-2 specific that were there at the beginning were there at the end. And the more samples you can take, the better in terms of getting more refined curves. And again, this is this comprehensive immune profiling by sequencing alone. And in the last few slides, I just want to bring this to the question of diabetes. There's several groups, maybe some folks that are here that are involved in this work in diabetes. So I'm showing, I've highlighted work from the Aaron Michaels group here, showing that there is a signal in people before they develop, when they're pre-diabetic, in terms of functional responses. So this paper didn't have any TCRs, but used those functional assays we were talking about. And then a couple of papers looking at T cell repertoires. And these graphs are from this paper that came out last year, showing specific responses to diabetes-associated antigens that were discriminatory and predicting either who had or who would go on to have diabetes. And you can see that these condition-associated TCRs, this sort of junction between the predictive ones and the non-predictable ones, isn't super clean, right? Like, so when you're doing these TCR studies, and this is true even in the infection situation, there's quite a lot of noise in the system, and this is particularly true when you're looking at single chain analyses. And all of these papers are single chain analyses. And where that noise comes from is, and I just have a few slides left, is the fact that if you're looking at a single chain, you're not actually recapitulating the specificity of the response. And a chain that you use in one response, even if it's highly characteristic of that response, paired with a different partner, is very likely to not be specific for your epitope of interest. And we have work coming out on this hopefully soon that show actually that basically 1 in 1,000 chains is actually complementary to maintaining specificity, even for a highly characteristic, highly motif-driven chain. Another source of the noise is the fact that the structure of the repertoire is defined by this power law. And when I've been showing you these power law distributions throughout the entire talk, where your very largest TCRs are much larger than the next largest TCRs, are 10 times larger than the next TCRs, are 10 times larger than the next TCRs, and so on down the curve. And so if you sequence only shallowly, you don't get much of the response. And you don't get very many clonotypes. And the response tends to actually, the relevant response tends to be actually quite low and quite rare. And so they're in this range of the TCR space, which you're missing entirely if you do something just like 10x type sequencing. That's shown in a different way over here. This is yellow fever vaccine response, but the bulk of the unique clonotypes that are induced are in that 10 to the -4, 10 to the -5 range in the blood. And this is in a response that's happening in the blood. And so in the last 2 slides, I want to present our current solution to this. So what we want in terms of an ideal technology for sequencing TCRs, we want something that's low cost, obviously. We want it to be high throughput, in that we want to be able to get into that 10 to the -4, 10 to the -5 range, and we want it to be paired. And our current toolbox is just these 2 approaches: the bulk TCR sequencing that I've been showing you throughout, that can get deep, 10 to the 7 cells, and is relatively cheap, $250. It's pretty tedious, actually, though. But it's also only single chain, so you're not actually recapitulating receptors, and it introduces a lot of noise because you get spurious associations with receptors that are appearing in different responses. The second approach is single-cell RNA-seq, so 10x or parse, where you can do 10 to the 4th cells, and that's 10x, $1,900 a sample, or 10 to the 6th cells, which is parse, which is about $20,000 a sample. And so I just want to mention, we put this protocol online, and we're about to update it again with some new details. But this is a new protocol we call TurtleSeq, which uses elements from SmartSeq 3 and then some novel approaches to make bulk libraries in a 96 or a 384 well plate in very small volumes, so in about 300 nanoliters. And then uses the co-occurrence of alpha and beta chains in wells across the plate to deconvolute pairs. And so as a result of this, for about $185 and 4 hours of hands-on time, you can sequence approximately 10 million cells, and get, in the preprint we show, hundreds of thousands to millions of paired TCRs. And I'm happy to talk in more detail about this, but the preprint is out, and the details of the protocol are all included in there. And so that's my time. I just want to conclude there. The work I showed today was largely from Anastasia Minervina and Misha Pogarelli. And then Misha is leading an internal initiative called Turtle that's driving this paired sequencing that we're applying to clinical samples to look for condition-associated TCRs associated with pediatric diseases. And our collaborators, including Ali, who I mentioned earlier, but I unfortunately left off this slide. But I'm happy to take any questions now.
Thank you so much. Please raise your hand to unmute or drop something in the chat.
Yeah, so Paul, thanks. That was a great talk. I have a couple questions. So one, in autoimmunity, we never have that prior to vaccination or infection. So having that true baseline gets really, really challenging. And I think it, at least in type 1 diabetes, is obviously different for every individual. So happy to hear your thoughts on how best to think about that, work with that, because we likely are sampling a relapsing and remitting disease over time. And sometimes we are able to, quote, see things that we know are potentially antigen specific, other times not. And then you're right, it's very noisy. And all of that work we've done too, I was hoping it'd be a little more black and white. So a lot of what we've been doing now is not so much bulk sequencing, but much more targeted, deeper sequencing, trying to look for that, clonal expansion, as you nicely showed on one of those curves of not a few 100,000 clonotypes, but much more in depth.
Yeah, great, and great to meet you. I love your papers. The first question is a tough one, and it's something we've thought about in terms of dabbling in autoimmunity. What's the best way to get these condition-associated TCRs? And I think your papers and other folks in the field have taken the two approaches that I think do work and can work, but are a bit trickier analytically. And so one is to just use people with and without the disease as your comparator group, and that introduces all kinds of noise because different people clearly have completely distinct repertoires. But and I think it's in one of your papers where you're looking at changes in flu or CMV specific responses versus what you're going to call the diabetes associated responses, and assuming that the diabetes specific responses should be changing and the other responses should not sort among the groups. So I think that's one approach that has to be HLA matched, obviously, to be super informative, and that's difficult. And I know there's some other efforts that are underway in Europe and in the US using really large beta chain bulk data sets, and using that kind of approach. They've had trouble because there's these populations, and again, it comes up in some of the papers I listed, are the emergence of type 1 diabetes is in children, and a lot of stuff's happening in children. Immunologically, they're getting a lot of vaccines, they're getting a lot of infections for the first times. And so if you're comparing them to adults, or if you're comparing them to other kids that are in a different part of the world, you're going to find condition associated TCRs that you learn your cohort, which some of that's going to be diabetes, but some of it could be other stuff that distinguishes those kids. Even some of the data that I've seen in matched siblings, which should get rid of a lot of that noise, you still have kind of maybe overfitting of some of the responses. And so I think it's tough, but I think it's at least if the same TCRs or the same TCR motifs start coming up in replicate studies, and you can map them back to antigens, obviously that's the final solution, where you can actually say, we know these TCRs are specific. The second approach is within the person, and if you have fluctuations over time in terms of the severity of the disease or the presentation of the disease, which I don't know how frequently that occurs in diabetes versus something like MS, where you might think that there are these fluctuations that you could sample across that would give you some indication of specificity. And then your second question was about, remind me what the second question was.
Yeah, that one was just maybe not trying to survey the entire repertoire, but now targeted deeper sequencing, which at the receptor level really helps you see clonal expansions in disease.
Yeah, so what we're doing with the turtle-seq approach that I showed you is you can pre-sort on phenotypes, and so if you knew activated phenotypes or memory phenotypes, or at least just excluding naive phenotypes, so that you're not sequencing naive cells and taking up real estate for no reason, I think that can be helpful. Clonal expansions are a good clue that something is drawing that out. And in our hands, though, you can see, you get clonal expansions that are not that they're spurious, but they can be sort of single data points, particularly if you're looking across people What gives you a ton of confidence that you found something condition associated is not just that it's clonally expanded, but that it's convergent in TCR space, that you have this motif. So if you see the same clonal expansion at the motif level on multiple people, then you have a really good sense that there's something common driving that. And then if you can kind of statistically prove that the most likely thing is the autoimmune disease or some other condition that those individuals have, that gives you a lot of, I think, reason to go after the target antigen. But ultimately, without the decoding and mapping back to a target antigen, it can be hard to have confidence that you have a true response. The last thing I'll say is just as an example of in a way how difficult this is, and where we sort of shifted our focus to trying to get the pair chain sequencing is, we generated this map for COVID, and we thought, we've been working on flu for decades, and COVID actually is farther along in a lot of ways in terms of the epitope space that's been mapped, and particularly on the repertoire side, the data that were available. And part of it was because we were able to get these true baselines on a lot of people. And so we thought this is the perfect test case for this as a model for dynamic tracking of responses. And I showed you some examples of that. But if you just tried to build a classifier of who had COVID and who didn't, and this, Adaptive as a CLIA certified test for this now that's proprietary in terms of what receptors they include, you think it would be super easy because we have just this list of TCRs. And it's actually really not easy, because if you just have the single chain TCRs, particularly because of some of that cross-reactivity, everybody looks like, if you just use presence absence as a metric, everybody looks like they had COVID. If you use abundance, you get a lot of false positives because you have these spurious clonal expansions where that receptors, I don't even think it's cross-reactivity. I just think that receptor is being used in another response, a flu response or an autoimmune response. And because we don't have the pair, we don't actually have the specificity. But we found if we could bring the pairing together, it cleaned up a substantial part of the noise. There's still noise from cross-reactivity. There's still noise from some other factors, but it's about 1,000-fold gain across whatever metric you're interested in terms of specificity by having the pair versus having single chain.
Thanks. I really appreciate it.
Thanks.
Any other questions, comments?
I have a quick question.
Yes, please.
Yeah, this is Tom from UCSF. Thank you so much, Paul. I learned a lot. And then I have a quick question about the reverse epitope discovery. And then I could have missed it. Do you require the candidate antigen, the continuous sequence of the antigen? Because in type 1 diabetes, we have these neoantigens that's generated post-translationally. And is there any issues with that?
Yeah, so it gets to the point of at what level of resolution do you want your answer? If you just want condition-associated TCRs, you don't have to know what they're targeting. And you can grow in statistical confidence that you found a condition-associated TCR, even if you never find the antigen, to the point where you're, pretty convinced, 99.995% convinced that you've got something that is a mechanistic mediator of the response to whatever you're targeting. That comes from looking at appearance across multiple individuals and convergence in TCR space and clonal expansion. And another element that I didn't get into at all because it's pretty technical, but you can actually determine for each TCR very easily the odds that you would have just observed that TCR or that TCR abundance by chance. And you can correct for that in your statistical models. And so that's called the generation probability of the TCR. And you can even do a selection model of that. And so that's something that's emerged over the last 10 years or so. And it's an incredibly powerful tool for correcting your condition-associated TCR observations. So all of that gets you the TCR list. But like I said, the gold standard would really be if you can then take that TCR as a pair and map it back to an antigen. So getting something like that myriad database for COVID was really helpful for us. And so I understand the complications in diabetes, and certainly in other settings too. we are working really hard on methods right now, and others are as well, to try to make that process of generating those kind of Myra-like data sets more high throughput, more systematic, and more agnostic to the antigens that you're starting with, so that you could pour in synthetic antigen libraries and start getting these associations. Because you need very little signal actually to make that platform work, when you're bringing in the statistical power of what you've learned on the repertoire side of things. And so that's, yeah, I don't have a great answer yet in terms of a universal solution, but I think we're not that far away from it. And so yeah, it kind of brings up this point of, where's this, there's sort of two models of solutions for decoding the repertoire. And so one is what I've just been talking about, which is this brute force empirical approach, where we're just going to map everything we can map through functional assays, and learning through condition associated responses. And then there's the sort of dream answer, which is going to be some structural AlphaFold 5 solution that lets us decode. And I think the field is sort of split right now, which one's going to win. And I'm on the side that we're going to probably, I think, the empirical approach that I've been talking about is feeding the AlphaFold and Rosetta Fold and structural based sort of, de novo theoretical approaches. And so I think they'll converge in time. But I think we're actually going to get a lot farther with empirical approaches than I would have thought of 5 years ago.
Thanks.
Thanks so much. Any other questions?
Yeah, if I could, this is Felix Braden.
Thanks, Phil.
I was just wondering in general, with the TCR work, how much you've been looking into the importance of the understanding of the germline T-cell receptor repertoire, and, do you take different approaches, or where are you?
In terms of like potential variation among individuals in the...
Well, and then also perhaps even, I guess what I'm really getting at, not only that, but then also how complete the T-cell receptor repertoire understanding of the germline databases are. Have you used different reference sets? Just something I've been looking at a lot lately. So
yeah, it's something it's something we pay attention to, but maybe not as much as we should. The bulk approaches that I was showing through much of the talk don't rely on that, because they're race-based. And so you're not using multiplex primers to amplify your response. You're sequencing off the constant region, basically. And that's true also of the 10x type approaches. If you're using a constant chain fixed primer, you should in theory see everything, and you kind of know you're seeing everything because you also pick up pseudogenes and non-templated sequences.
Sorry, I was thinking more like during the annotation step, when you annotate the full sequence and figure out what's your V gene, what's your V allele.
So do you think it matters what so again, when you're doing the bulk sequences, you get the full length of the sequence. So we annotate it to the closest annotated sequence, but if there is a variation, you capture it. In the 10x approaches and in some of the other multiplex based approaches, including adaptive, you don't get the full length sequence. And so you annotate to one of the databases, and then if you try to then make that receptor, you fill in what the database tells you that receptor is. And that has introduced in problems for us and for other people, because there are polymorphisms and or the annotations are wrong.
Right.
But we tend to rely on IMGT and others with sort of limited updates on our ONN when we're convinced we found polymorphism in our population or individual that we're looking at.
Okay.
Hi. Thank you so much for a great talk.
Thank you, Maki.
Oh, sorry. Can you hear me now?
Yes, we do.
Thank you so much for the great talk. I'm Maki Nakayama at the Barbara Davis Center. So I have a question about self-antigen versus pathogen, virus antigens. So probably because the affinity to self-antigens may not be as strong as, affinity to pathogens. So sometimes we cannot cluster our TCRs, even we have both alpha and beta chain sequences. Clustering are not as strong as what we can see with, for example, like flu virus specific TCRs. So I was just wondering if you have like, experience or something that, you are facing on with self-antigen specific TCRs studies.
This is a great point, and I think it's a pretty subtle point actually, because I'm not totally convinced it's affinity or self non-self per se. I think it's more about and it might be affinity associated, but I think it's a pretty loose association. So where we've addressed it more, because we don't look at autoimmunity as much, but we do look quite a lot at cancer associated antigens. So cancer associated and cancer specific, so neoantigens, but are very close to self.
Mhm.
But even within viruses, you see some responses that are not the ones I showed you. We always show these ones because they're they're how we started the process, and they just scream at you. But they're super immunodominant. And so it's more about what's immunodominant versus what's not immunodominant. And I don't think immunodominance is necessarily affinity on the TCR peptide MHC side as much as it is on maybe the peptide MHC side. So I agree, a lot of really relevant, biologically important responses are not as pretty as the ones I was showing. And as they get less and less pretty, less and less immunodominant, less and less reliant upon high generation probability receptor pools, it's harder to see those clusters. And so that's where kind of pushing the clustering algorithms has I think a role to help us. And so I think our sort of simple, fully sequence-based cluster metrics are working, and they can work down to some of these harder responses, but there are some responses where they definitely break down. But we assume there is actually some similarity in if not in 2D sequence space, in 3D structural space. And so my hope is that as we, I think we're very close to sort of cluster-based methods that rely on structural information, even if the structural solvers right now are not good enough to just sort of de novo tell us what the antigen is. If you tell a structural solver, I see these sequences associated with these antigens, then they actually can give you some information. Yeah.
Thank you so much. Thank you.
Thank you. Anyone else? Let's see anyone in here.
One last question, Paul, are you interested in type 1 diabetes and working on self-antigen T-cell receptors?
We definitely are. If you're, we're always looking for collaborations, and particularly with the turtle-seq stuff I showed at the end, if you want us to run some samples, we're very happy to run samples for folks. Yeah.
Great news. I just had two very quick questions. Mine are very basic, but one before we head out. You showed that the viral phenotypes in context of SARS-CoV-2. Has anyone else done this work, or use these methods in context of pediatric CVB infection? Is there like a pre-existing data set anywhere that you know of?
DMV or CB? What's CVB?
Coxsackie virus B.
Oh no, I don't know anyone that's done Coxsackie. That's interesting. Yeah.
Okay. And then the last question is hypothetically, if you took samples from those at genetic risk, pediatrics, patients that are at genetic risk for T1D, and then sort of just following the pediatric vaccination schedule, do you think you could determine, could you see any TCRs using your methodology against islet epitopes?
I think it's possible. I think that's what's going on in some of the work that I showed. And I know there are groups working very hard on this. I don't think it's not a screaming signal. I think if it was a screaming signal, they would have found it. But
Right.
As we get deeper and as we get better, the signal has to be there. It just is on us, I think, to figure out how to tease it out.
Yeah.
Yeah.
Get rid of the noise.
Yeah.
Okay, thank you so much. This is fantastic, and we look forward to host or having you join us again on the TCR working group.
Sounds
Thanks.
great. Thank you so much.
Have a great rest of the day. Bye-bye now.